தாடியெல்லாம் ஏன் கழிச்சிங்க? அது போதும், அது ஆடியோ கிடைக்கும்ல, அது போதும். இந்த இதுவும் போட்டுருங்க. இதுல cyclooxygenase symposium சொல்லி ஒரு ஃபைல் அதுவும் வச்சிருக்கேன். அவ்வளவுதான். Yes, sir. We have a quiz in the afternoon. You know that, right? The online, no, right? Did they come? People come in. வீடியோல ஆடியோ இருக்கா? வீடியோல ஆடியோ எது வீடியோல ஆடியோ இருக்கா? ஆடியோ இருக்கா? இல்லைன்னா இல்ல ஆடியோவும் கிடைக்கல அதுல. சரி இல்ல இல்லைன்னா அதுல ஆமா அதுல கேப்ஷன் தான் கேப்ஷன் ஃபர்ஸ்ட் தான் சார். எஸ் இல்ல இவரோட கொடுத்தாருல. ஆமா சார் அவரோட சார். நான் முதல்ல கொஞ்சம் இன்ட்ரோடக்ஷன் Can you hear me? Suprabhadam, good morning. Wonderful to see you again. So we have pretty much people that we are expecting. We'll start. I think I should not be waiting for the people here. A conscious crowd with purpose would come before the speaker comes. I was here well in time. You're not in time. Something, a problem. And it's my duty to remind you that's a problem. Why? Things continue to be in state of rest or of uniform motion until an external force acts on it. All right. So with a little bit of admonitive message, I start the day. This is the last day of our symposium. All right. So thank you for being a part of it and being an active part of it, most of you till now. So we'll start off with, until now, what and all have we seen? Can anybody recollect? I want it to be an interactive symposium, right? So what have we seen till now? We have seen some basic chemistry, some simple reaction chemistry. Then we have seen basically how these catalytic schemes, how they convert into some electrical activity, how they convert into some sensing response mechanisms, how they convert into some powering homeostatic mechanisms. Like for example, in xenobiotic drug metabolism, the hydrophobic molecule gets converted into hydrophilic molecule and the water washes it away, right? Simple logic of how, without really asking for too much of specificity and all that. So now we are going into the even more complex aspect of how, from a molecular level, we saw some electrical, now electrical and mechanical, right? Macroscopic, how phase changes, how things like that occur. So we will, have you heard of this bacterial flagella? What's it called? It's called bacterial flagellar motor. All right, I want to tell you that this was subject of a major book by Michael Behe, who said that this is an example of irreducible complexity. What does irreducible complexity mean? Something like, if you are having a machine that's more looking like a man-made machine, right? The assembly of which takes systematic, very difficult arrangement which cannot spontaneously self-assemble on its own, then it's called irreducible complex. Multiple components are needed, and it is highly improbable that it could have evolved on its own. All right. So now, yesterday's casual interaction, we had Mohammed from Charusat saying he was upset that he was taught something wrong, and all the efforts that he took is going down the drain, right? So the reason I put Carolyn Wells' statement as the first and the foremost, what was it? That critic is necessary and criticism is a necessary evil and all that. And then bracket, I had put that if something is not right, it should be discarded soon, and it should not be continued because it is not good for the society. We cannot continue to train our youngsters on wrong paths, wasting resources, time, all that. That's not good. That's not what the hallmark of human civilization is. So, with that message in context, I want to introduce to you a new addition to our School of Artificial Intelligence at Amrita, Dr. Biswajit Das. Please come onto the desk. So, Dr. Das is primarily trained as a chemist, and he shifted on into computational mathematics and all very sophisticated aspects, the kind of works that you see in papers and you don't understand, right? Very sophisticated work. Only a very small fraction of human society would be understanding that. So many of the ideas that we have are basic physical, basic chemical, basic visual ideas that can be easily tracked. But if it becomes abstract, too much abstract, and delves into the complications that you see in mathematical texts, it has notations and equations and things like that. Somehow it's away from your day-to-day life, and that kinds of, you don't get interested too much into that. Only a small portion. So he is among the small portions, the intellectual elite kind of people who understand the intricacies of mathematical modeling and computational simulations, and like Prasad, right? So you people, now the point is here, Prasad has been working in this system, bacterial flagellar system. And he has also worked on potassium channels, modeling and computational aspects of that. So, so I thought it'd be a good thing. He has been working as a postdoc in China, all right? Peking University. It's a prestigious university, is that right? Yeah.
Rank is 39. World rank.
World rank, 39. All right? All right. So he was there, and his wife is a faculty at IIT Palakkad, so he wanted to have a job here. So, and our dean, he looks to recruit multidisciplinary people. I have no bearing with artificial intelligence, yet I am a faculty of School of Artificial Intelligence. So now one thing is that research is highly interdisciplinary. If you are from bio background, please remember this thing that I will only study this much. I'll only like to add gene, cut things and put it somewhere. No, that doesn't work. No, the world is changing. This is a fundamental message I wanted to give you. So now I give the dais to our Dr. Biswajit Das, who will talk to you about what he did with this bacterial flagellar system. At the end, he has prepared some slides. Basically, how bacteria are mobile. Until now, we were seeing how they metabolize chemical, and then how they sense response, how they maintain their basic components and things like that, chemical, then chemoelectrical, things like that. Now we are seeing mechanical aspects, how they navigate. All right. So are you going to provide any historical perspective or something? No. All right. Whatever you want to do, you take this because, our Joshi put it I'll switch it
Okay.
on. See, it's
Okay.
to be pressed for some time, and the light comes.
Okay.
Hello. Yeah,
Yeah.
it's on.
Yeah.
So all yours.
Yeah. Thank you, KMM sir. Yeah. So this is the yeah. Okay. This is pointer and this is the change slides. Yeah. Okay. Yeah. Hello. Yeah. Thank you. Good afternoon. Good morning, everyone. So actually today I am going to tell you about. Oh. Okay. 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 Oh. Okay. 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 Actually, today I am going to tell you about that I am why I am interested in murburn theory. Yeah. So because I did during my PhD, yeah, I did work in chemical physics related works, especially on ion channel and energetics on non-equilibrium energetics on ion channel. And after actually did my during my postdoc, yeah, I shifted my field in mathematical biology. So where I started this field, bacterial motility, and then actually I some questions actually arise in my mind. So after joining here, yeah. So when I discuss with Professor KMM, so I understand. Okay, so I can actually use this Marban concept in this field. So I am not going details about my work today. Yeah, little bit actually I am explaining why Marban concept. I am interested in Marban concept as an introductory lecture. Yeah, so that's why you can understand. Yeah. So and for that actually I will discuss about the traditional concept of bacterial motility, and because I have to tell. Yeah. Otherwise you can't understand why Marban concept and where actually Marban concept is needed. Yeah. So I will explain. Yeah. So hello. Yeah. So bacterial locomotion actually is an assembly of run and tumble motion. Look about the this is an YouTube video. Oh, yeah. So look, this is actually bacteria actually moving forward. This is actually it is called run. And look that bacteria. Oh, sorry, I am playing again. Yeah. So look, that bacteria is actually in pausing mode and it is changing its directions. Yeah. So it is called the tumbling motion. So now, so again, you can see this is the run motion. Bacteria is moving forward and this is the tumble motion. So it is making some pause. Yeah. So it is called tumbling motion. Now, if you see, well, oh, it is not. Yeah. So okay. Yeah. So if you now see, so this is the run motion. So where actually bacteria moving forward, and this is the tumble motion where bacteria making some pause movement. It is called tumble. So from experiment, it is actually I am telling you the conceptual, the what is actually now going on, not in the Marban theory. Yeah. So, and in this field, Professor H.C. Berg in Harvard University, he is very pioneer in this field. He did his lot of experiment on to cultivate this field. Yeah, and he actually observed, and he maximum actually he did his work and on that field. He suggests that during run motion, so this filament. So if you see, this is the bacteria. So this is the cell body, and this is the hook, and this is actually the filament. And overall, it is called the flagellum. Yeah. So when during run motion, flagellum rotate in the CCW rotation, anti-clockwise rotational, counterclockwise rotational direction, and in the tumble mode, it is the flagellum rotating in the clockwise direction. And this is actually how actually you can explain this run and tumble motion. So you can explain is so this is a cell body. Okay. So now I am coming the concept swimming on bacteria. You can explain to the resistive force theory of hydrodynamics. I am coming to the concept of mathematics. Yeah. So see, this is a one only monoflagellated system. Yeah. So only one flagellum you can see from, you can consider for simplicity. Now if you see, this is the cell body, this is the helical filament, yeah, is there, and now it is rotating. Yeah. So basically bacteria moves at a low Reynolds number. Do you know what is a low Reynolds number means? So low Reynolds number indicate whether it will be, the media will be turbulence or not. At low Reynolds number there is no turbulence. Yeah. So bacteria, you can, I am not going details about the hydrodynamic concept or about that. Yeah. So it is usually you can see the Stokes regime. Yeah. So you can, and if you see, it is the, oh sorry, 1 minute. Yeah. So this is actually rotating in the counterclockwise direction. If you consider it is rotating in the counterclockwise direction, then and this is the radius of the filament. This is rotational. This is, through this radius it is rotating. Now you can see, you can consider one small segment. This is a small segment of the filament. Now this is the radial velocity u. You can consider u equal to omega into r. Omega is the angular rotation and angular velocity. You can understand. Can you understand that? Yeah, this is basic physics. Yeah. So now it is called. So you can just divide it in the vector just notation. You can just decompose into two parts. This is parallel component and it is perpendicular component, and it is making some angle theta like that. Say this is a theta. So this is u cos theta and u parallel equal to u sin theta. Now according to resistive force theory, you can see that in the Stokes regime, the repulsive force, yeah, so it is not acting just opposite to the u. It little bit makes some angle f. Yeah. So if you decompose this, so you can parallelly you can decompose in the two part, f parallel and f perpendicular. Then you can see there is one resultant component ft. You can write in this expression f perpendicular cos theta, f parallel into sin theta, and also you can see this is one another component is there fp. This is f perpendicular sin theta minus f parallel cos theta. So this actually this force is the propulsive force. So this force helps the bacterium to move forward. So if bacterial flagellum, one flagellum rotate in the counterclockwise rotation, from resistive force theory, you can see there is one component. This component actually just pushing the just pushing the bacteria. Yeah. So that's why bacteria moves forward during run motion. And for that actually, this is actually it is called a symmetric type of motion. It is, so that's why as filament rotating in the counterclockwise direction, so cell body rotate to oppose that reaction. Cell body rotation, cell body rotate in the clockwise direction. Yeah. So this is the general concept of the hydrodynamics. So now, so this is a reference. You can actually write these references. This is a very nice paper. Eric Lauga-Elter. He is in Cambridge and he is in Department of Applied Mathematics. He is working mainly in this bacterial hydrodynamics. Yeah. So now I am coming. So if you see the in the run motion, so you can understand there is, it is called each and every filament actually producing one propulsive force. This force is acting that direction. So this filament also acting that direction. So if you see that just vector calculation, you can see there is some net propulsive force definitely acting in the forward direction, and for that bacteria move forward. Now if you see in the tumble mode, Fv actually this propulsive force acting in the opposite direction and in this direction. So this is this is just so. And when actually it is rotating in the counterclockwise in the direction, clockwise direction. In this rotation, it is counterclockwise, and this is the clockwise direction. In clockwise direction, if you just actually simply you can just make some calculation, you can understand that the propulsive force definitely acting in this direction. Yeah, so in the opposite direction, you can prove it. Yeah. So that's why actually FP acting in the different direction, and there should FP net should be 0. So that's why tumble motion happens. So this is actually the simplified version of the hydrodynamics theory. I am telling here. There is another lot of theory are there, but I am not coming details. So and also observe it is from also from experiment it is observed that during the run motion, this hook become soft and rigid. Sorry. Oh, sorry. I am making one mistake. Hook becomes soft and flexible. Okay. And so this is actually, you can see this is a Brown et al. Actually did this experiment. So he is also belongs to the HC bar group. And during tumble motion, hook becomes straight and rigid. So that's why actually this FP net becomes 0 and acting on the opposite direction. This is an explanation why run and tumble motion actually happens. Now I am coming the more actually microscopic. I want to give some more microscopic description. So say why bacteria actually this filament actually rotating in the counterclockwise or clockwise direction. So say this is the run motion. So this is in the counterclockwise rotation is happening. Now if you see this overall flagellum structure, then you can see this is the bacteria. So this is the this flagellum actually composed with one filament, and it is connected with one bacterial flagellar motor. Yeah, and it filament and BFM connected through the bent hook. It is actually bent remains CCW direction when rotating, and in CW rotation it becomes straight. So now why actually this rotational transformation occurs? So now if you see in the tumble mode, then it becomes straight, the filament becomes straight. Yeah, and it changes in its rotational direction. So this is actually due to the reason of the one phosphoregulator signaling protein CHYP. It actually binds at the lower portion of this motor. This is usually FLIM and FLIN protein. I am not going into detail because my next speaker will give details about the structure. Yeah. Okay, and KMM sir will describe. Yeah. So about the molecular structure of the BFM. Now it is actually the traditional concept. Yeah. So this signaling protein actually binds to the lower portion of this BFM. Yeah, and that's why this CHU, the rotational direction changes. Yeah. This is the concept. Now, is the motor torque and speed is measurable? So AC Bark actually measured this rotational speed and torque. I am giving the experimental details little bit, yeah, just introductory level. So suppose this is a multiflagellated bacteria. So now, first actually it truncated all of the filament except one. Yeah, so except one. So then it actually emerges in the viscous medium and is strictly attached with a glass cover sleeve. Yeah, so it emerges in a viscous medium. Then actually you can see this one bead, latex bead is actually attached with this truncated filament, and you can observe that the bead is rotating. Yeah, the rotational, you can observe this rotation of the bead. So why BFM rotates? Because they actually think that BFM, this bead rotates as actually this filament, and the filament is attached with this bacteria flagellar motor. So as BFM actually rotate in the counterclockwise direction, that's why actually bead is rotating. Yeah. So this is the experimental setup. So how they can measure the motor torque? So this is you can see that this is the revolution motion. And now motor torque is calculated as, say, this is the motor torque, and this is the rotational drag coefficient. So drag means rotational drag coefficient means, so if you actually rotating, if this latex bead is rotating in the viscous medium, yeah, so it faces some friction, yeah. So this is actually frictional force. Actually, it is called you can think the rotational drag coefficient, yeah. So the rotational drag coefficient of motor, and this is the load. It is actually called the in experimental language, you can say this is a load. So this size of the bead you can vary, yeah. So and you can get the different speed. So you can get the omega for different values for different bead size. This is the experimental technique, yeah. And whereas zeta L actually you can measure, and this is the RB is the radius of the bead, and eta is actually the viscosity of the medium. This is the standard technique, yeah. So it is described by H.C. Burke in actually Harvard University. So you can see this is the reference. Now, surprisingly, if you actually, so you can measure, you experimentally you are measuring omega, and then from that equation you are calculating tau m, torque. Then actually, if you actually plot omega m versus tau, then you can see for CCW rotation, so there is one plateau-like region. Then after one certain phase of one speed value, is certainly this velocity goes to zero. It is called the knee speed. However, in presence of the CHEYP, this signaling protein, this omega relative torque versus speed value decreases linearly. Yeah, so there is some asymmetry. So actually, I am not going the details about that, but this problem remain unsolved. Why it is occurring? So I solved that problem. Yeah. So it is almost 14 years old problem. I solved that problem. But actually I considering this the traditional concept, not murburn concept. Yeah. So now I am not going in details. So now actually this is actually the high viscous loads. This is actually medium viscous loads, and this is the low viscous. So low viscous load means the bead size is small. Yeah. And the high viscous load means bead size is very big. Yeah. So now if you see, so this is asymmetric in nature. Okay, 1 minute. Now if you see recently, you can see one picture. Oh, so yeah. So you can see this is Simogonia et al. Actually did this experiment. You can see torque induced precision. So yeah, you can see bead is rotating, and it is actually making the precisional motion, not only revolution. Yeah. So you can see this is the precisional motion. The bead is performing precisional motion. So that's 2015. It is published in scientific report. Yeah. So why? Yeah. So you can see this is the precision is observed. You can see this is the so precision. You can see from physics concept, precision is the resultant of the revolution and spin motion. Yeah. So precision actually observed. You can see the revolution as well as spin motion is observed. So in my work, I also considered these two type of motion, and I explained this asymmetry. So now actually this truncated filament and bead exhibit precision motion. Now a question is that in this case, so bead is, according to the traditional concept, BFM is rotating, and that's why bead is rotating. So BFM actually rotational of due to rotational of the BFM. So spinning motion of the spinning motion, you can observe the spinning motion of the bead. Yeah. But why actually you can see also the revolution motion. So why it is actually happening. So you can actually also describe to the hydrodynamic theory or vector theory. But now I can see that molecular mechanism of torque generation. So if you see the BFM, this is the filament and this is the hook, and here this is the FLIGC protein is there. So it is concept, and this is the stator. Yeah. So H plus ion mainly enters from periplasm to cytoplasm through the stator. Yeah, and it contacts with the FLIGC protein and makes a power stroke. What is power stroke? Power stroke means just a push like that, this type of force. This is called a power stroke. Yeah. So, uh, according to this normal concept, actually, that what is the torque generation mechanism, I am explaining little bit. So, stator can stay in the two conformational state. One is a stator without proton, and another is a stator with proton. So two proton can enter into one stator protein, and there is 8 to 12 non-rotating stator. So 2H plus when actually say this stator MOTA and MOTB protein, actually KMM cell will explain later rigorously about that. Yeah, regarding this concept where actually murburn concept is needed. So I am actually explaining the what is the concept is now in literature. So now actually, so 2H plus, so there is two say piston like that. Yeah. So this is MOTA protein. One MOTA protein actually when H plus ion comes, it just moves onward and it down. Yeah, moves up and down, and it makes out to one power stroke. First power stroke, and this is the second power stroke, and it pushes the FLIG protein like that. This rotation, and that's why this rotation of the FLIG motor, actually FLIG protein or rotation of the motor is happens. Yeah, this is the concept. And when CHIP protein actually binds to that seeding complex, it changes just the direction of the FLIG proteins. It changes. This is the structural actually study. It done by Paul et al. It is published in PNS. You can see this the direction of the FLIG protein is just changes. It's changed. Yeah. So if you see the structure of the this direction of the FLIG protein is just changed in the direction, you can easily explain the CCW and CW rotation. So this is the concept. Now you can see why CCW rotation. So this is the I stator. It is making one this force is there, and this is actually the Z axis. So you can see from the torque relation, you can see this is the tau. This is the stator rotor interaction. This is the torque is acting along that direction. This is the radius, and this is the force. So it is working. It is rotating anti-clockwise. And now if you see in this CCW direction, just you can explain it very simple just vector model. Yeah. So now you can also just its direction is changed. Now question is that now I am coming. So why Marban? This is actually the very brief summary of the already existing theory and literature, and why actually I am actually feeling that why Marban concept is needed. Because see, I already explained that 1 minute. Yeah. So H plus ion is coming from periplasm to cytoplasm in both rotational modes. So H plus ion always coming inside. But in literature, there is no such, actually, I don't know, according to my best of my knowledge, actually, there is no mechanism how H plus ion, so H plus ion is entering. But if H plus ion always enters into the periplasm to cytoplasm, so pH will change. Yeah, inside. So how actually bacteria maintain its pH? So I think in this regard, yeah, so this is I already mentioned. So if and for that perspective, actually to get the answer of that, yeah, I think uh, if I use the Murburn concept, I can explain how bacteria actually maintain its pH if H plus ion or continuously entering into from periplasm to cytoplasm. So this is actually and the reason that's why I am interested on Murburn theory. But later actually KMM sir will describe details about the already existing concept, where is the drawback or where is the blah blah blah. Yeah, so you can you can listen. Yeah, so that is I think for that is enough. Yeah. Okay. Yeah.
Yes, thank you so much. Oh yeah. Can you hear me? Hello? Yes. Okay. So, how they, thank you so much. So, he gave the classical, contemporary, thank you. Okay. So, he gave the Professor Dr. Das gave the mathematical explanations of sophisticated interpretation of bacterial flagellar motility, flagella assisted motility. Okay. Now, I'm going to shred whatever he said into pieces. He is, why? Because he spent a lot of time building upon so many things that was already built. And the kind of sentiment that he expressed, that is, when we don't check, before we do a work, what did I always do? Explore, resolve, prioritize. All right. So mathematicians and chemists and physicists, when they get into biological system, biology is highly information centric. Yes or no? Too much. This name, that name, that name, this name. Physicists go, nah. Mathematicians go, like that. So now I'm pretty confident what I'm saying is true, but I'll keep my mind open regardless. Let's get some terminologies clear. Rotational. What is rotation? Repeated circular movement of the rod along an axis that passes through a center of symmetry on its own body. All right. Then revolution. The body traces a circular orbit around an external locus as its center. A stone tied to a string forced to swirl around. Spiral or helical, the movement is around an eccentric locus, but the space-time coordinates are not periodically repeated in exact terms. For example, a flower or something falling down like that, right? That. Torsion, right? You go, comes back, some spring assisted or some movements like that. So I'm just getting these words very clear. What is it's called? Paul Boyer's explanation is rotary ATP synthesis. There is torsional ATP synthesis mechanism. There are many kinds of proposals. Okay. So now these kinds of, but in natural, in spontaneous living systems, Howard Berg, whom he mentioned, he passed away last year. Good for him. Because it would have been sad for him to live to see this. He was a good scientist, was a meticulous scientist, but then sad. So, ways to rotate a rod. I'm not an engineer, but when you see how can advection, see your rod, fluid can be applied. It can flow like this, the rod will rotate. Okay. Then direct torque, you apply something in, and then you can rotate that by virtue of that will. Then gears, you can connect it to gears, gearing or belting, you can have it move. So these are the ways to rotate a rod. Then yet there is yet another way to rotate a rod, that is by electromagnetic induction, right? How you have the motor generator, that assembly, basically magnetic field, and you use bushes and all that, and then you rotate it. This is the simplest way. Now, I want you to see the bacterial flagella. I want you to see where it falls. All right, these are the tangible, real ways. Now, proton has a charge, it comes in, But is it mechanical? Is it electrical? Is it chemoelectrical? Is it space? How does it happen in real space with respect to real force? How was something that people saw an observation? How was it interpreted? And how was it put with respect to real concepts of physics? And I'm a biologist. I don't know all these sophisticated things, but this is the structural aspect. So GPCR, transducin, you saw in rhodopsin, that was so big. Complex 5 is so big. This guy is huge, humongously big. All right, and it spans multiple phases. 1, 2, 3, 4, 5, 6, 7 phases, at least. All right. So, you know, this is the way you could get a simple thing to basically rotate. Basically, is like that, simplest experimental way. Now, what Dr. Das showed is that this is the rod, this is the membrane, this is the C ring, flea proteins. There are flea G, flea M, flea N, lot of number proteins. I'm not going to bother you with the names, but I'm going to ask you to see certain things. Now, let me just go first before I come back to that slide. I want you to see one thing. You know, there are left-hand screws and right-hand screws. Yes or no? You know what? How they differ? You know how they differ, right? Yeah, one, you will drive it in like this. One, you will drive it in like this. All right? There are two different things in real space. They have different directions. Now, you must understand that when you have a bacterial body like this, if you have a right-hand screw, the very, very elementary thing is that a left-hand screw rotated counterclockwise cannot push. It can only pull. The structure of the bacterial flagellum observed in some have a screw-like shape. Not all flagella. Some flagella are flaccid and they just are like that. Some flagella are like a screw. But they are comprised of polymers, monomers that arrange in a left-hand screw kind of a, when they self-assemble, they assemble sometimes like this in a particular polarized manner. Now, a left-hand screw cannot. In 1970s, somebody published this and the whole world did not even bother to check this fundamental aspect. Did you understand what I'm saying? This is very fundamental. Left-hand screw can only, when it can push, it will go forward like that if it is clockwise. But the observation is not that. Observation is that the left-hand screw is pushing by counterclockwise motion. So that is not real. That cannot be real. Next, mechanism of bacterial flagellar motor. It's supposed a motor. Now, when it is counterclockwise, these are the actual counts of protons. We have already seen this, right? A bacterial maximum without accounting for the space occupied by macromolecules, very few protons. Practically, a micro-dimension bacteria doesn't have free protons inside. Now, this course, his question, he said that he's interested in murburn concept because how does it maintain pH? My question is more fundamental. My question is more fundamental. The idea that it rotates was fundamentally supported by Peter Mitchell's theory that the protons are pumped out and cycled. There was no question of pH regulation. Did you understand? There was no question of pH regulation. It comes from periplasm, Cytoplasm pumps it to periplasm, periplasm to cytoplasm. There was no need for anything. It was this is how people change over decades without the generations knowing what's the problem. They will change this. You are witness. I'm telling you, they will change it. But they will take jolly time so that all those people sitting at the top, they will have no problem. They will save their faces without getting any shame or anything like that. So why? We make mistakes. We can accept it. What's the big deal? I also taught such things. I also did such things. But anyway, so the proton, there was no question of, this is not good for me. My neck is already, no, wait, I'll take it. Okay, I'll hold it in my hand. This protons were actually pumped out and they were coming back, like in mitochondria. Now the problem is, there is no proton. Now, the uncoupling method is that the uncoupler basically dissipates the proton gradient. This is the logic of. In 1970s, earlier the theory was that ATP was assisting. Then they said, no, it is just a proton-based idea. Now, I want you to see very clearly. Proton is what? 0.00002 angstroms. And sodium is what? Around 1 or 2 angstroms. Vibrio cholerae can use protons as well as sodium. If you have a machinery that can rotate with 0.0002 angstroms as well as 2 angstroms, my goodness, what a machinery it would be. Right or not? All right. So, you see the uncouplers. With the uncouplers only, they changed that only protons are needed. No other mechanical or all that is needed. Just this proton, or later on as they say, seen by. Why? See, some bacteria, they are in what pH? They swim around even in pH, alkaliphilic bacteria, pH 12. There is no question of proton there. pH 12. Kahan? Nahi. Then they said sodium. But they didn't think about the size of the ion. Right? Are you getting it? You don't need to be a rocket scientist to understand this. Alright? Now, uncoupling thing, the proton. Now you see the data. Applying data with a global perspective is very important. If you try to say, now you see pKa. If you want to study biochemistry, pH, pKa, 50% biochemistry, you understand. 50% of biochemistry you understand. How to make a buffer, what does a buffer do? You understand biochemistry. Practical biochemistry. Now you see, these are, so this is dinitrophenol. The IC50 is what is given in the black. Nanomolar, micromolar, nanomolar. The structural aspect and the pKa aspect do not seem to be affecting the inhibition. If it is proton and all that, you would expect. And then there are molecules which don't have free protons, but yet they are serving as uncouplers. The point is to reason out all the kinds of data. This doesn't. And most interestingly, this is just a, structurally if you analyze, it's a secretory system. What is a secretory system? Something that puts something from inside to outside. So for example, have you seen the video of or a pictorial rendition of how bacterial positive, the sexual conjugation, it has to transfer its DNA from inside, and so it has to put something outside. So it's what started something like that, later on the toxins, all that, when it puts into the host, that is done through such secretory mechanism. So it is a type 3 secretory system. What does the secretory system do? It puts some fluid or solid from inside to outside. And most importantly, the structural aspect, when you see, here you see these multiple layers it has to process. The whole shaft must rotate, and this must be leak proof. And protons are what dimension? 0.0002. And it has to rotate. How can it rotate? As well as, I told you in the cytochrome P40 area, kinetic isotope effect, it was rotating as well as binding. Rotating, I want to have the cake and eat it too. Protons cannot come in through any other place, but they have to come in through a particular place like this. Now, before 1990s, something else was the rotor. Then they found that, they mutated. We'll come to that part. Now, here is murburn view. Murburn view is water goes out. There is a lot of hot water produced here that goes out, and this gives an apparent feeling of rotation. That's it. And how does it work? What does a flagellar do? You have seen a kite, right? Have you seen a kite? Kite, if you try to get it upward, a small kite, it goes round and round. Fluid dynamics doesn't work properly. You put a tail, it goes straight. Small bacterium, if it pushes water out, it will have only tumbling. So you put a tail to it, it will go in one direction if that tail has some preferable dynamic properties. Now, if you consider this left-hand screw thingy, the classical model fails, and also this has to rotate, this has to rotate. It's a continuous thing. The whole thing has to rotate. No, so the torque can only give very minuscule thrust. Even Howard Berg says it's only a 1% efficiency system. All right. If it has to go round and round, it will only give 1% of the energy, that is. Because you see, it's a very thin thingy. And the load on this because of the length would be too high also. Now, I'm not going into the mathematical aspect. I want this video to be run. Let us see if it... the video... Yes. Please see this. I'm not going to comment. Can I get back to... Okay, what can somebody tell me? What did we just see now? What did we see now? Yes. Yeah.
Flagellar alone can rotate itself without the rotor.
Yes, but there was water coming there, right? Yeah, water was coming. It did not need a rotary thing. And now, one basic thing about your sense of reality. If something is tethered, attached at one end, will it rotate? What was it showing? It was looking like it is rotating. Yes or no? So, the whole observation of rotation could have been a mirage. Now, when you see a dog, no, but there is a lot of video wherein that bead tied and all that, lot of things. I will come to that. It's a complicated thing. It's not that easy. But I want you to understand, when a dog licks water, tries to drink water, it does like that, right? What do you think it is doing? You think that it's scooping water like this, right? But it's not doing that. What it's doing is it's doing this. We think that it is doing that, but it is actually doing this. It's not pushing it towards its mandible, towards its upper jaw. It is doing it towards its mandible, lower jaw. So that's a big difference. Big difference. It's not really rotating around its rod. But then we see in certain cases when we attach the bacterium with the filament, you see the bacterial body rotates as he showed. But that rotation is only around 10 per second. What these people are claiming is that the flagella rotates thousands or more. Thousands to 10,000. It falls there. Yes. Now, these two are two different things. We'll come to it. We'll come to that discussion. I know you have worked on it. I know it is hard. I know. So, the big thing is, now, this rotor, so-called rotor, requires protons to come in through particular places like this. So, before 1990, this was the actual rotor and this was a stator. Now, this is the rotor. It changed. Why? Because they saw some people mutated the amino acids that were supposed to be allowing the protons to come in. They mutated all the amino acids and they saw there was no effect on the rotary motion. So they shifted, they said this was the rotor. Now what? And they said there was a D32 that was conserved. One amino acid equally that was conserved across all these systems, they said. And that amino acid helped proton come in, something has to come in. That was what was allowing its mechanical movement. Now, I would imagine if they said so, it is true. All that we did is Danny cross-checked. Now, you see that D32. Some people don't have any D or E, even 5 or 6, 4 or 5 amino acids, this side or that side. What does it do to that mechanism? You see, multiple D's, E's. Absolutely spurious claims to back up. I'm saying this on documented. This is being documented. They said this is conserved. How dare they say that this was conserved? It's not conserved. And then the Fle, all these proteins that they were supposed to be a sophisticated mechanism like this to move clockwise, counterclockwise all that, if it is actually a rotary motion, you would require, can you imagine how this is? It is, yeah, not only that, how precise the articulation has to be. If it has to be rotary, it has to switch from this to that side. It's unimaginable. And how can they move? He put a picture wherein that movement like a jaw, that ratchet movement. Now, how can that work when there's such a huge force there, and you try to move here? When you switch on, even in the highly powered electrical system, we have in our house one fan switch. You switch on, it thinks, should I move? And so what I'm saying is, the inertial tug is so much with one. How? What is the rationale for that electrical, mechanical, or whatever force? They say it's some kind of spatial idea, and then their electrical idea. There is no real tangible way of transducing it in real space with respect to the mass and all that. They just say, they just use some equations. How does it happen in real life? Nothing. All right. So, and then the protein that is supposed to shift is this shifting, this flagy. You see its structure. So we saw the structure clockwise and anti-clockwise. There is no way it can articulate like that and shift like that. For us, it doesn't appear. So fundamentally, we find multiple flaws in their aspect. And also, if it is something that rotates, you would imagine with such thing, all this, there should be some conservation. You see the number of amino acids, the sizes of these relative things, they're completely different in different species. This kind of an evolutionary mechanism must be crazy. You try to make something that rotates, try to do it on your own. How difficult it is. This cannot. So now I forgot to put the image. I'm sorry, I forgot to put that image. Our theory is that the whole thing here is based on collecting reactive oxygen species, making water. And so this must be highly polar. I had that image. I should have put that image. That one image would have answered so many things. Like, you remember sodium potassium pump? That how it was having so many arginines there. Here, all that it has is highly polar and charged, packed like this here. So what it does is water, superoxide, peroxide, radicals, all that focus, make water, heat, boom. And there is a ATPase like switch here, which is a mechanical switch. ATP hydrolyzes, water goes out. That's it. It's a jet propulsion kind of a thing, but not taking from outside and pushing it. That, what was his name? Low Reynolds number, Purcell put because bacterial flagella at this, they are in low Reynolds regime. In low Reynolds regime, inertial forces cannot act. So, bivalves, what do bivalves do? They take water, for example, octopus, for example. How does it do? Takes water, boom, pushes it out, and then moves forward, right? That cannot be possible in this. But what is happening here? It is not taking water from outside and pushing it outside. It is making water inside and pushing it outside. And there is one lab in Caltech that has given the math for this. So, clearly, what does the flagella do? It stabilizes the fluid dynamics. And that multiple types of flagellar structure are there. So, you imagine if it is rotary movement, how will different kind of flagella, how will they accept? Very simple, water goes. Some things can go like that and they can assemble. It is not always helical. Some are very flaccid also. So the push and pull, and most importantly, this, there is no beating this. There is no beating that. As Sir said, there was nothing to rotate it, but it was rotating. Second point, it was attached at one end. How can it rotate? There is no scope. There goes the theory of mechanical rotation. What does it do to science? You have a tangible way to understand evolution now. Right? How can something move? Suppose a chlamydomonas, it doesn't have a brain, but it has light coming in. So the light is coming from this direction. How can it choose to move there? When the light comes there, ROS gets produced, they get water, and then it moves, it continues to move in that direction. Did you understand? It's a very simple logic. Very simple logic. There is no need for a brain to decide to move, how to go. The tumbling motion that he talked about is when water goes out, it stops. So there is a misbalance of forces or something, and then it cannot maintain that direction. And then once again, water jets out. That's my interpretation, right? My interpretation has no flaws till now. It is not proven either. But the current interpretation is definitely has got multiple problems. Yes, and for me, when somebody works a lot of time, and then somebody questions that, it is not a pleasant thing. Nobody wants to do that. Nobody wants to see their lifetime activity going into waste. You don't want to waste your time in something that was wrong. So when I don't want to talk about it. So somebody, one of my students, it's painful. He started doing this PhD project on this. I said, this is a falsely framed question. Don't do your work on this. One second. So he did it, but anyway, he also bought it. Dr. Das also thinks that ideas are good, but then there is a significant amount of traction on that.
Yes, I want to discuss.
Yes, we will discuss that later. Now, so it's your discussion. Your discussion now. Go ahead.
Yeah, no, actually, I strongly agree with Marwan theory, but actually, I have some need some discussion regarding the rotational concept. Yeah, because so precessional motion, you can see the precessional motion is observed, and I also mentioned one paper, Professor Eric Lauga. Yeah, he just written in, it is published in Physical Review later. It's a well famous journal. So he actually showed that if for our shear flow, if in a shear flow, so filament actually making polymorphic transformation, and he mathematically proved.
Yeah, we saw that video.
It is also at a polymorphic transformation.
No, but that is because the flow.
Yeah, exactly.
The flow can change it. The flow can change it. That's
exactly.
what we saw.
Yeah, exactly. So that's why actually a lot of discussion and lot of hydrodynamic study. Still, actually, I suggest Professor KMM that we have to perform a lot of hydrodynamic study to establish that. Yeah, and another actually I suggest that okay, we have to need some lot of discussion about the experimental evidence like that. Yeah, so if it is possible, so for H plus moving out of H plus. Yeah, so this is. But I strongly believe the Marwan concept that. What is how actually bacteria actually maintain this type of pH? I just tell in terms of pH, yeah. But already this answer
the point is when the field came, we should do this.
Exactly.
And people should see that.
Exactly. Actually, this is actually the hybridization of the new classical and Marwan concept.
Yeah.
I think so. And
there is no hybridization.
No,
there is no hybridization.
No.
The classical was wrong, and the new is right.
No, I have a new theory.
There is no concept of hybridization. Yeah, because here, when we have a theory, it has to build ab initio and it has to be placed in proper foundation. And that foundation is when you try to. Now, can I take one second? A speaker comes to a classroom and takes a jug, and puts 3 big stones in it. All right? This is very important. This is a very important story. Stories everybody can understand. Data and mathematics people can't interpret. Stories everybody understands. Very simple. So he takes a jug, puts 3 stones in it, the jug fills in. So he asks people, can we put anything anymore in this? So people say no. Then he says, wait. He takes smaller stones, puts it in. Then he says, can we put any more in this? Then he takes some sand, puts it in. Then he says, can we put any more in this? He takes some water, puts it in. Now he says, try to extrapolate this with respect to your life experience, your approaches, all that. So some kids stand up and say, you think you can only do this much, but you can still do more. One good thing. So many people give so many interpretations. He says, if you take it as a problem solving or philosophy towards life, what does this experiment just that I did, the demonstration that I do now, what did it show you? That is, you have to put the larger stones in first to solve a problem. To get the approach in your life, you have to prioritize, explore, resolve, prioritize. Here you have a problem. It's a jigsaw puzzle. There are conflicting observations. First thing that you have to put is, what will you put? What is the most important thing that you will put? Such a problem. Assembling something across 7 faces without leaks. How can something be rotated by protons as well as by sodium? What is the dimension difference? 10 to the power 5 to 10 to the power 6. There is no scope for the other theory. Absolutely no scope. And then these are proteins. And then counterclockwise moving, all that energy yields nothing. It's a simple thing. The structure, secretory system, very clear. They are just assembling, sealing it. It becomes tight enough. Water goes out. We have seen the other experimental data. There is no need to discuss anything any further. As far as jettisoning the older idea is concerned, it is wrong. Hook, nail and sinker, it is wrong. But regarding the new idea, there is a lot to be discussed. Because, for example, we don't know how does the water come out like this. I have just shown this. Water comes out from here. That is what is to be dealt. And I don't know. I don't have the facility. I don't really care much now for this. For me, the problem is solved. For me, it is disproved. I've given a new avenue. For me, that nuts and bolts. But that is what is important. That's what runs life. But the selfish guy that I am, my challenge is to, there's some intellectual kick I get and I move on. That's it. That's bad actually. That's not great. No, that is not great. For me, until it is useful to people, it is a worthless idea. It's just an esoteric pursuit. Life is all philosophy. There is some path as well as a goal. Both path and goal are essential.
Very important. If you have been stuck to a particular one problem only, then other problems could not be attended.
Yeah, that is true. But that is true. That is true. But for me, yes, it has to. Yes, I agree with that. I agree with that.
Somebody else can be
some idea you got.
Yeah.
I just leave it halfway for somebody else to do. Is not a
good way of doing it. Absolutely, I agree with that.
It is not about your selfish pick also. It becomes a responsibility once you take it into the world that this is an idea.
I agree with you 100%. Yes, that is why recently I showed you that the first slide that I put, Albert Szent-Györgyi, he said there are scientists of 2 kinds, Apollonians and Dionysians. Now Apollonians, they systematically they fill in the gaps and take students, get funds and they do that. Dionysians like me, boom, 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 they go. Now, both are important. I'm not negating the other one. But I want the Apollonians to... Yeah, they have come? That's come? Yeah. So the tea is come. Sir has come? Sir, no? Okay. So the tea has come. What is the time now? 10:54. Yes. Now, yeah.
Sir, as you said, many people are working in a field with the classical knowledge and classical R&D they are doing and wasting.
Pay attention, please. Discussion is where science is.
And many have wasted. They feel that they have wasted their time and energy and money. So basic understanding of the science. That science is a part where the mechanics, physics, chemistry, mathematics, and mechanics. If this would have been understood better at the lower level of education, undergraduation, then further research could have been stopped by themselves. If there is some mechanics problem is there, or there is some mathematical problem is there. So the basics to start with a PhD or research is to fully understand at undergraduate level physics, chemistry, and mathematics.
Correct.
So these three subjects must be studied in three years. We did. When I did a food technology, the course was designed as it. If you don't do BSc mathematics, physics, and chemistry, you will not get admission to the course.
Yes.
So it was very easy to learn other things.
Yes, choice. Yeah. So my as far as learning is concerned, I think the most important subject to learn is what, Mathematics. Yes, our dean loves mathematics and all that. For me, stories. When you are kids, the stories you learn, they are the most important thing. Hans Christian Andersen, Raja Nanga hai. That concept. Pied Piper of Hamelin. People are led. Rats. That what is shown is that when I was young in first class or second class, I studied a beautiful story. Three fishes. They are in a pond, Kerala medium school, and once they're happy, they're living in a pond. Everything is nice, resources. So some guys come around and say, "Hey, come on, this pond is filled with fishes. Let's cast a net next weekend here." So one of the fishes, overhears that. And don't ask me how the fish heard it, okay? Don't put your rational mind here. Okay. Story is a story. There are some... Yeah. So, yes. So, fish, yeah. No, but language, I said the other side also. Hathi chalayega kaise? So, yeah. So, the point is that the fish goes and lets the other person know, other two fishes know. He says, "Hey, look, I heard this. They're going to cast the net. We better move out." The second fish says, "You know what? I heard it. I think there could be some purpose in it. But I like this place. Let's see. It comes. Aega, dekha jayega. ठीक है? Then this third fish says, "बहुत देखें. क्या है यहाँ इतने साल से जीते आ रहे हैं. We have been living here for so long. Nothing's going to happen." That weekend comes. That people come. They cast in it. By that time, the first fish has already taken a small canal. It has gone to another water body. The second fish is a street smart fish. What it does is it pretends that it is dead. Floats on top of the water like this. So the people take it and just throw it, in Kerala local. So it's in a pond nearby paddy field. It also falls in because they don't want to consume something that is dead for what reasons, So third fish gets caught, gets fried, gets eaten. Moral of the story. Stories are here to teach you basic approaches in life. I tell you, the fundamental message that you should take from this is chance favors the prepared mind, explore, resolve, prioritize. Pursuit of excellence is based on what you have and what you know. These three messages. Not major things that you learn in textbooks. All this. My life pursuit has been based on these three philosophies. One second, sir. आप बीच में वो बोलेंगे जब फ्लो में रहता ना आदमी को गाड़ी चलाने दीजिए. एक मिनट चलने दीजिए. जब फ्लो में रहता ना आदमी को चलने दीजिए. आपको समझना चाहिए. नहीं. Yeah. So, do you understand that? Because when Narasimhan sir showed me and said, something like that. That, he builds on what he knows. Krishnadas, I built on what I knew. When I prioritized solving that stones, then chance favors the prepared mind. It was not said by me. Who said that? Pasteur. You know Pasteur? Yeah. Our bio people's core. So, these people have built their lives on fundamental philosophies that they have learned through stories that you do learn when you are young. So, the younger you are, the better you can learn. Because otherwise, our learning. So, I think, yes, you wanted to say something, I will go.
In our curriculum, we used to learn a lot of stories based on Panchatantra and some based on the yoga, Vedas and all that. So Rishi Munis were knowing that these are required to learn by the people at a younger age. Very important. And our forefathers or our relatives, which are elder at that time, they used to tell us the story. Lot of stories from Pancharatna. We do not have to read at that time. Say 5 years, 3 years old, we do not have any chance to read. But they used to tell. So if you have a joint family with the father, grandfather, everybody, then this thing works. And now that is been killed. So people are not able to learn the stories.
And the ability to identify what is the crux of the story is also nobody knows.
That intelligence has lost, and now people like this so-called modern people, they don't want to go into the intricate details. Why the story is told, even that is not that is lost.
We used to go to his home in the vacation and just stay 1 month with her and to listen the stories the next. Lot of stories were said. Now where are we? That is lost. now people are going.
Yeah, please have your refreshment.
Hard reality.
Hard reality. We get only, and now people don't need stories. People have visual. Your nerves are all visual. Quick. Yeah, please go ahead. Use that. And then we will. Soman sir को बुलाते हैं यार.
We converted into some presentation.
Where's my phone?
To the level of global problem. These small things, somebody
We don't use that please, because I told you.
it can solve the global problem of all global problems. Basic problem global but क्या है? What is basic global problem? Fighting. Each and every country is fighting each other. Somebody is putting the bomb and all problems. बहुत छोटा technique. लेकिन again it is the basics of the biotechnology. Basics. If you don't know this, you cannot enter. I'm not a biochemist there. But I have learned meeting people like that. Important is learning how to stop. रात को prepare कर रहा हूँ.
Yeah. Please go and come back quickly, and we have to take picture. Sir, I'll call sir. Hey, 1 second. The number of people, Muttu, you have to let the people know. Number of people who are staying in the hostel. 32. So we will say around 30. 35 சொல்லலாம். Night 35. 32 தான் வருதா? One second. This is something more important. I said 10 o'clock, 35. Those girls who are staying back, they need food arranging for that. I don't know if I can arrange. Because there is a 12-hour window that they asked. Ramapan. Super. Thank you.
What I learned was, basically I started learning. That learning is required. I understood from it. Learning is required that I learned from it. That you require learning. And I was very thankful. Everything was lost in 1 hour.
Yeah. So this is the thing.
Ego, entire ego was lost.
The ego base of ego is also very important one. So that is
more, but sometimes we miss
yes,
and learn the life of people, understand the language, extend. Then my learning will be that because my doctor was my senior doctor. It's not giving somebody else.
Why? Why when you say why we get more knowledge?
Yeah, so I understand something is left. I just repeat it for a year.
Because the moment you share it, it goes and your heart and mind is going to look forward for the next thing. If you are keeping it with you, you are developing vision that you have the knowledge. You are on the edge. You are putting yourself on the edge.
I think
yes, you learn. I want to.
Aging is a problem.
That is different. That is
but I will not invite anything.
yes. So I want to. Yes.
Why we can do? But this is another problem.
I said.
Where we can. You are from here. I'm from Mumbai. Sometimes I come here. Other places here. We don't do it. Yes, thereby just 4 kilometers. More time.
why can't you live in symbiosis? We are living in symbiosis. Yes, that is it. Or else this disease you need to pinpoint. There everywhere in the area. What is the minor breakdown of it? Simple monomolecule protein or anything. That is very important. And how we can
Ma'am, you said you have to go there and you came back here. Oh, some money is flowing out. Are people inside? What happened then? Do this. What is being asked?
നമ്മുടെ നാഷണൽ ഇന്നോവേഷൻ ഫൗണ്ടേഷൻ ഓൺലൈൻ അപ്പൊ അവര് ഇത് കാണിക്കും.
team should come so that to take a picture. He's bleeding. So that's
So can we take the picture? Eh? How do we take the picture? Oh, look at him, he's sweating. സാറേ എങ്ങനെ വന്നു അന്നത്തെ വന്നു സാറേ ഒന്നും പറയായിരുന്നില്ല സാറേ വണ്ടി സാർ നടന്നു വന്നു हमारे टीम के चल के आए उधर
Please. We are going to make a group photo. I need here. So we'll all go together. Let's not anticipate. Let's not go missing and all that. Quickly, we go. Are we going to the front? We are going to the front, right? Left. We go to the front. To the portico. We take a picture there quickly. And sir has to leave quickly. Let's come all the way from there. So we'll be nice. We'll go quickly and we'll come back quickly and start the message. Okay? Please go the way they take you. Please go. Hi, Aileen. Hi, Aileen. Are you there? Oh, she has switched off her mic. In a content, so okay. Anyway, support. I try to reach her. So message I came up. Is it on? Hello? 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 Oh, yeah. Battery, come here, sir.
बच्चों को तो हम समझा सकते हैं।
नहीं सबको समझाना चाहिए, जिधर जरूरत है उधर जरूरत है। हेलो,
Yes. So the remaining program is like this. We are now into a filler mode with Danny and I substituting. Actually, the talk was about the summation of murburn concept, but then we'll have it perhaps at 3:30. Yeah, we'll switch off this. We'll close this door so that nobody comes from there. So yesterday I talked about this sodium potassium ATPase, and we'll just do something regarding that. And I will just give you a broad eye view of where murburn concept is. And then today, we just, in the morning when we got up, I saw biological chemistry had rejected our paper on cyclooxygenase. Wonderful rejection letter. Smooth, sweet, no nonsense, very cute. So I wanted to expose you to all aspects of research. So that will come later. Danny will, I'll quickly in about 10 minutes, I'll cover some aspects that I missed out regarding electrophysiology yesterday. So now, very fundamental aspect is that now you see what is written here. Can you see what is written? The first few italicized papers are about the foundations of the experimental foundations that led to murburn concept, and the remaining are the applications of murburn concept in diverse fields. So that's what the papers are in decent journals. So this thing, if you have, you can always follow what murburn concept is. This is solid. I do not need, do not need any further endorsement in respect to publication and all that. Publish, done. Nobody can get rooting in society only if you people do something about it, because otherwise it's not going to happen. So with that, let's just quickly get over the remaining slides and then go to Danny's content, and we will break at around 12:30 for Professor K.V. Venkatesh talk. He has assured me that he'll come. He's swarmed in meetings, but he'll come. 12:30, he's now in Australia, so it will be evening for him. So otherwise he had promised to come here. So now this is what regarding this guy, pumping and binding logic. I told you that this is the protein. So now you had these molecules, that steroidal molecules. They were supposed to, do you remember the structures of those molecules? And now you see here, the kind of inhibitors that they are, modulators that they are for NK. You see, it goes all over the ballpark. All over the ballpark. Okay? Steroidal, non-steroidal, some molecules and all that. Now, the point is, all these are redox active, interfacial redox active. But sodium potassium ATPase is not a redox enzyme, right? It doesn't have any cofactor. It is just hydrolyzing ATP and pumping sodium potassium here and there. Now, how do all these affect it? These can all affect it because of the murburn theory. Murburn theory says that it is based on oxygen's redox activity, which moves the potassium in and sodium out because of these things. This is the reason why we have that sieving. You see, potassium, if potassium will be inside in the presence of electrons, it will be minus 19 plus 3. Inside, sodium will not be that preferred because it is plus 2.1. Did you understand that? No? Yeah, you understood that, no? Yeah. It is the thermodynamic reason. It's not because of some willful pumping. Do you understand? No enzyme can do that. Okay, so then I wanted to show you here miscellaneous. It can explain simple structure and oxygen sensitivity, uncouplers, how they affect observation of ATP synthesis by NKA. So what they did is, there was Freeman Cope and John Glynn. John Glynn was a Cambridge biochemist. He won. He said ATP is synthesized by NKA, reverse of ATP hydrolysis. But then the synthesis is because it recruits ROS. Do you understand? So misinterpretation. Then the transdermal peptide. I told you something, that structure you see, lysine, histidine. Okay. So this kind of, I told you in cytochrome P450 that N term had lysine, and that messes with the ROS dynamics. So then these diverse molecules, and see the phosphorylation aspect. This phosphorylation is seen on so many places. Here, there is a phosphorylation here also. How can the protein get phosphorylated there? That is a transmembrane region. If it is by ROS, it can. Not by an enzyme. Did you understand? Did you understand? So the phosphorylation is not just seen. What they said is that, see, the binding of ATP, then the binding, it gets phosphorylated. So they said when it gets phosphorylated, it has got affinity for potassium. When it is non-phosphorylated, it has got affinity for sodium. How? Mazak hai? It doesn't happen like that. The thing is, the phosphorylation is not just occurring at one place, multiple places. That is a stochastic phenomenon, because once ROS is there, it can transfer a phosphate to any OH group. So this ADP has OH group, that's why phosphate gets attached. The serine, threonine, all those also have hydroxyl group, that's why phosphate gets attached. No enzyme can go into this lipid filled region, but a small radical can go. These are, kind of supporting evidences, for the new theory. So I also, you see, the assessment of if this, I had shown you one gate opens, then potassium goes in, then closes, then something goes out, then that gate open closes. Even if you basically, for non-selective and reversible process, the overall possibility of this series is 1 by 800,000, whatever, the huge number. Even if you have a selective and higher binding value of half, the overall score would be only 1 by 16,000. Because everything is reversible, because same molecule is doing the process. So that is what I have shown here. And what the questions at the outset. Okay, no reason provided why ion permeability changes during various phases of action potential. Why? Why should they change? Potential change, the protein gets permeable different. The protein makes the potential. It doesn't make any sense. So many writings, this Farachi's writings, Tasaki's, Terakawa, all these people's works were completely sidelined, cherry picking data, not making global sense. So this is something, and the pumping thing, you get the cellular in to out pumping and out to in pumping. If you just put your common sense to it, you will see that it doesn't work, because all that this 3:2 achieves is, one, sodium getting pumped outside extra, right? Now, what will happen if it constantly pumps like that? All the sodium from inside will get pumped out. And what they say is, no, there are also channels to allow them to go back and forth. So it is like making a picture for you which does not explain anything. one guy goes to a shop and he buys, he takes some 10, 12 banana. Look at me. This is more important. 10, 12 banana, one gucha mein. He takes and he says, Bhaiya kitne ka diye? How much for this? He says, 30 bucks. So he says, okay. With the other hand, he takes 2 oranges. Ye kitne ka? How much for this? Roughly around 30 bucks. Okay, he keeps the banana down now. He takes an apple. He says, how much for this? One apple is around 30 bucks. Acha, then he puts the orange down. Okay, then starts biting the apple. Apple ka paisa jiye 30. I put orange. Orange was in lieu of that. So orange maine liya nahi. I didn't get the orange from you. Or he says at the banana. Orange, I put banana. Banana ka paisa. Banana maine liya kahan? Where did I take the banana? There is no real accounting. It is just pushing one thing to there. When you ask, like the mirage that MC Escher drew. Did you understand? At one point, one thing substituted for the other, but on a global scale, nothing explained. So that's what this, if you also see the dimension. So for one cell, if a macrophage changes the dimension, the surface volume ratio and all change. How does the system account for that? How does? There is no logic to account for that. You understand? Suppose a cell is there. Have you seen a macrophage change its shape? It can change volume to surface area very dynamically. How can it maintain its concentration when it doesn't make sense? So these are the kind of things that I wanted to show you. And I also wanted to say that murburn concept is also very important for diverse protein modifications. The hemoglobin glycation and all that. It's because of murburn activity only. All right, it's a redox process. So basically, like murburn modifications, you can also see in non-accessible areas. So this is the elaborate scheme of murburn post-translational modification. Okay, so the zeitgeist is science advances by negation, society is served by correction. We have to have multiple aspects, global data points, thermodynamics, kinetics, structure, function, all that must appeal. Only then it becomes a theory. So with that, I give the stage to Danny. Around 12:30. Aitcha. 15 minutes is enough. Cyclooxygenases. Do it. Yeah, you can do that. Come. Why are you not doing? Professor, we are waiting for Professor Venkatesh. So.
What are the energetics involved in modifications of a, is there a unit loss which will create this much of conformational change? Can that energetics be derived?
No, we have not done anything like that. What we have done at this moment is, Prasad.
It is more of observation.
Yes, rational, qualitative reasoning.
Qualitative rational explanation has been given.
Yes, basically, now we are observing so many things, right? Now, to all the phosphorylation, all the glycations and all that, there are so many possibilities that can happen. Only an enzyme-mediated phosphorylation possible. A ROS-mediated phosphorylation also possible. Similarly, glycation, it is also possible. So, sometimes when what biologists generally do is protein structure, when they do not understand certain things, when they do not have direct tangible explanation, they always resort to what? And lo and behold, conformation change occurs. That's a very, sometimes very relevant explanation. And then affinity changes. These are the two reasons. But then why should a conformation change occur? Why should an affinity change occur? These are important questions. Prasad asked me, what is the energetics? In the sense that, have you followed the exact sequence or accounted for something went in?
My simple understanding. I am from a structural background.
Yeah.
I believe in that, and some structures have to happen, and the structure provides the infrastructure or the basic mechanical support system for the chemistry to happen where ROS may come in. I am very happy for that. Okay, but your ROS system is already happening. But to enable that ROS system to work or murburn concept to work, my question is that how much of this ROS to conformational changes the energetics are related. So what I mean to say is there is an enzyme. Suppose a kinase is there. A kinase, one flip happens, and that flip makes one active to inactive. And in this, suppose, for example, hypothetically, a ROS is involved in that. So how much ROS is needed for how much of conformational change?
That depends.
So
that depends. You would imagine on where the
unit of and because if ROS is playing a role in ATPase movement, because my what is that called excitement in ATPase molecule is, I'm coming from an engineering background. So when I see a motor and a stat and struts and all those things and a gear system, I get excited, and I have worked for past 25 years on that. Now, yes, that particular infrastructure might be there, a support system is there, and your ROS is also maybe complementing it that in some other way. But where is that energy coming from? And is it that completely we are disallowing the concept that there is no need of energy? It is just raw species which are coming and then going off.
No.
And so how do we? There is.
I get it. I get it. So what you mean to say is your approach is from the perspective that structural information of the protein is what allows catalysis, right?
It is enabling catalysis.
Yes. Now here is something. Now the other aspect is the energetics part of it, the overall aspect.
Which has to support the whole theory.
Yes. Now the point is that now when you put oxygen into any system, even at room temperature, there is spontaneous formation of ROS with electron sources here. There is. So it's an experimentally ratified thing that there is effective charge separation occurring. Oxygen is able to receive electron. So it is, even in room temperature, it is a facile process. And the chemistry of electron transfers of this is well established. So the feasibility or the energetics is good. Now, how much exactly ROS is required to denature or transform or protein,
yes,
that depends upon each specific protein, I would imagine. So now if you have a protein that has a very crucial amino acid.
Sir, simple question. Simple. In your own analogy, I am traveling from here to there. The velocity is 2 meters per second. I am not traveling 2 meters, but per second I am traveling 2 meters, right? So similarly, how many?
No, that's what I address. I understood that question. That is what I'm addressing. Now, if you have a protein like this, you want some change, a structural change to happen in this protein. And if you believe that ROS mediated phosphorylation or some hydroxylation or oxidation, something that could happen, what is the energy term that is required? How much ROS would correlate to a protein getting modified or changed? Is that?
I don't have that vocabulary for that. How many ROS molecules will give me 1 angstrom or 0.5 angstroms of conformational change?
I understood that question. Now, the point is, now, suppose if I do this, अरे यार गुदगुदी तो होना चाहिए तेरे को. My example का बेड़ा गर्क कर दिया. So you touch him in the right spot, he'll react, right? So that's what I'm saying. Now the protein has a particular conformation. It is energy minimized with respect to the surroundings. Now that if you can perturb at a particular place, it will get affected more in response to some other thing. Or
say there are hotspots.
Yeah, there are hotspots.
Yes.
So now the point is, your question is how much of ROS or what is the total energy term for n number of ROS to affect something tangible that can give some kind of structural change or something like that. It depends upon protein to protein. It depends on ROS. It depends upon environment. It will depend on that. That's it. It has to be. It has to be.
Planck's constant is there.
Yes.
It is universal.
No.
Pi is there. It is universal. Similarly, for these many rods, or for single rods, this is the change.
No, you did not understand one thing about ROS. ROS, how many ROS are there?
Yeah.
Superoxide, singlet, hydroxyl, peroxide, all these.
Yeah.
So now and then
are you able to, in your mechanism, explanation of mechanisms based on murburn theory, okay, are you able to identify which ROS is actually responsible for that particular? Most likely, yes.
And I can't really, because when you have superoxide, they dismutate spontaneously forming peroxide. And then peroxide and superoxide can react. You get hydroxyl radical. Or they're all in interactive equilibrium with each other. And the question, I think your question is from a physical chemist perspective, or more the structural engineer's perspective of a protein.
Exactly.
Yes.
I would like to, because I have seen things happening in that way. You are telling about big molecules here, I mean nanomachines, which I teach them as nanomachines. Okay. So you are talking about nanomachines, but what I have proved in a patent draft is that even engineering a particular simple monomer like aspartic proteinase, 325 amino acids, it has various layers of machine components. So I identified them and how they relatively move to each other. That is creating the function. Okay, there in this particular scenario, most likely I don't know whether ROS is involved or not. So there may be a boundary where ROS is effectively acting, and that is the explanation. Or if ROS is playing a role in this also, how that particular, I told you that 7 angstrom are the lysines or the charged polar amino acids in a particular protein are able to create this ROS spontaneously and result in that particular function to happen. So this particular concept. So my very purpose of coming here is there a way I can further apply or further contribute to this murburn concept, and that's the main thing which I'm interested in.
Yes, I want to show you some things. Like now, a protein is stabilized, energy minimized with respect to substrate. There's a serine here, there's a glutamate here, it enters a hydrogen bond there, something like that. It is like that. Now comes a raw species, attaches a glycate molecule or a phosphate molecule or something. That interaction is perturbed.
Yes.
Now it depends upon where the protein is, what kind of protein it is, which part of the protein has been randomly modified. That's what I'm saying. So
in that case,
because we are not controlling where the ROS goes, but now the future of murburn is, now we saw the sodium potassium ATPase, right? The protein will have a ROS and ion dynamics through it or near it. That will be a probabilistic route. Now those probabilistic routes, if they are altered, if they are modified, if they are messed up by those diverse kinds of molecules that are shown, then you will have impact. So it is, now I keep saying this, there's a mountain there. If you put some water from the top, it will come and get accumulated in a pond or some other place at a lesser height. It will follow multiple routes. This is the whole point. When ROS reactive species, diffusible reactive species is involved, we have always looked at cellular processes with only the structural aspect of the protein. Here what we are looking at is something that can affect the structural aspect, but there is an interactive dynamics of molecules, the ions and the reactive species that can also affect the overall outcome. The protein affects something on its own, then this interaction of these things outside the protein or around the protein can also affect. And the protein can also get affected. That's why I showed in this post-translational modification.
There is a chance that there is a skeleton. As much as you are passionate about ROS, okay, and nature's simplest principles, nature also has its own simplest way of doing things with respect to the creation of a skeleton for a particular mechanism.
Yes.
So in that case, where is ROS play in collaboratively working or symbiotically working with the structures? So that also That is what my intent.
Yeah, I understood. The point is that Ross dynamics is what allowed the evolution of proteins. Because, or Ross, no, diffusible reactive species. Because fundamentally, they are the ones that are energetically driving things. Because if you see the cellular fundamental energy pathways, 2 electron processes, they are always around the glycolysis and all that. They delta G 20 kilojoules here and there.
How much of light from external source can influence the cellular internal mitochondrial cell walls in me?
Well, I was talking about the photodynamic therapy yesterday. There, when you
this light is influencing, and that means this light has the ability.
And I also talked about how one mitochondrion kept here.
Correct.
Yeah, because it's a high energy reaction. That's why.
So
that's what
that
ultimately.
The whole macroscopic energy is influencing the nanoscopic or picoscopic.
Sure. Regimes.
Creating oxygen. This redox. ROS.
Yes.
It is creating. So that means any disease, that is disease, can I control by this type of external? I am thinking.
Wonderful. That is our paper we had sent to Cell Reports. Exactly, that's what I'm saying. Now here, Lauren Jekyll and I, we had put down a paper wherein this is a system, interactive system, wherein it can generate radiations and it can use radiations. So there are cancer cells that lose their tumorogenicity by changing its location, the kind of the contacts it has the kind of radiation it gets, all that. So matter can affect, can produce radiation. Radiation can interact with matter. In fact, that particular thing is being shown in the last talk that Laurent will give. That's the final talk. So basically, your question that how do the energy term interact with structural aspects, right? That is what you are coming to, right?
In a way, but there is a lot of synergistic aspect also I am expecting, just not disregarding the nature's structural build it has done all the...
No, I'm not disregarding it at all. I'm saying I'm complementing it. I've never ever No, I clearly stated that in multiple occasions. We are not negating, just because I've discredited certain affinity-based theories in certain models, I'm not saying that there are active site, there are no active site, there are no structural effects.
So you are understanding that particular aspect very clearly. So why don't this aspect be synergistically explained about this bacteriophage, about this ATPase molecule, and about the flagellar?
Oh, sorry, I did not explain the ATPase molecule. We have a paper on that. So bacteriophage, I have no clue what you're talking about. What I want you to, I think, Prasad, what is happening here is, we are not saying there is no structure-based outcomes. Of course, very well established it is. What we are showing is a completely new interactive dynamics that we have overlooked. That's all. And that can also affect structure, that can also produce radiation, that can also, there's a huge window of opportunity to understand that. That's all. I'm not negating anything, saying that there's no movement and all, but then without movement also we can explain. That's what I'm saying. So sometimes certain things are not understood. People invoke only conformation change and affinity change. There are things which are interactive dynamics also can explain. That's all. So 1 second, our Professor, has he come? Vandana, has he come? Has he come? Has not come. So Danny, you start. If he comes, yeah. 1 second, we'll take the interaction later. We have enough time. All right. Yeah. So Danny, you go ahead and speak, Danny. We'll take it because Danny's lecture is prepared. So this is contextually very important because we worked on this. Have you heard cyclooxygenase? An enzyme called cyclooxygenase? No?
Yeah.
You have COX. It's called COX. COX1, COX2. We have kind of studied the whole mouse, human, COX1, COX2, all that. So you will not be happy. Certain things that we say now. Yes. Yes, that's what. We are also complementing only. That's good.
Okay. Like Sir said, these cyclooxygenases, perhaps one of the most important enzymes in the drug industry. Because you see that there are certain omega-6 polyunsaturated fatty acids like arachidonic acid, which is the primary substrate of cyclooxygenase 1 and 2. Cyclooxygenase 1 is known to be present in all tissues and it has got some functions, right? Some homeostatic functions. And it's kind of expressed all the time, you can say. It's there for some functions in the cells. But cyclooxygenase 2, COX2, is inducible. It's an enzyme that can be produced when it is necessary. So you can see that arachidonic acid is metabolized by this enzyme. This enzyme is like the Roman god Janus, as we have said, two phases. It has got two active sites. One is a more lipid-buried active site. Next slide. Okay, there is a more lipid-buried active site which is oriented towards the membrane, where oxygen is known to go in, and two molecules of oxygen are utilized per fatty acid. AA here represents your arachidonic acid, whereas the other side, diametrically opposite almost, you got another cavity which is very large. It's like a cave, I would say. There is heme right there. We have worked on so many peroxidase systems. We very clearly know that murburn reactions occur in peroxidase systems. Now we just needed to translate that into the cyclooxygenase system, because what we have here is the active site is buried, arachidonic acid is liberated by phospholipases, it just has to go in and then it has to get converted and come out. But there are some fundamental questions that we want to ask. So all of these, what you see here is that the ion goes through different redox states, and then there is a generation of something called a tyrosine radical. I want to keep it very short. This tyrosyl radical, especially tyrosine 385, is generated. And that is the agent which drives the catalysis, they say. That, if you go back to the previous slide, we can see that there is a carbon 13 where attack happens, and it converts into, see, what you have is like a linear molecule. Of course, there will be some kinking because of the double bonds, but it's like a thread. It goes in like a thread, but it comes out more like a hairpin, right? So you got PGG2, which is the product which is formed from cyclooxygenase site. There are two sites, cyclooxygenase site and peroxidase site. So the next product is this PGG2, which is generated from the cyclooxygenase site, gets liberated, goes all the way around according to the conventional mechanism, and it enters the peroxidase site and goes and binds there. And it abstracts 2 electrons from the heme center, which is there in the peroxidase site. And then PGH2 is the product which is generated, and downstream there are other tissue specific enzymes which make products such as other prostaglandins as well as thromboxanes. So prostaglandins are mainly the pain hormones which you have, and the thromboxanes are involved, for example, in platelet activation. And so prostacyclin kind of does a function where it can prevent clotting and things like that. It's a good thing, but there are bad, mostly those other hormones which are generated. Now you go to the next slide after this. So if you see the structure of both cyclooxygenase 1 and 2, if you see the active site, if you see in this A diagram, the green thing right there in the center, I don't know whether you'll be able to make it out, in the crystal structure of a particular rat cyclooxygenase, arachidonic acid is bound there. So you can see that all these amino acids, they say, are involved. There is some kind of van der Waals force that is able to accommodate all that, and there is a particular arginine 120 at the active site mouth which will stabilize the COO minus part of the arachidonic acid. So what we can question is that the rate of oxidation of arachidonic acid is far better than many other polyunsaturated fatty acids. If it's the same kind of mechanism where a particular active tyrosyl radical is involved, why would that occur? Why should there be a disparity in the oxidation rates of various fatty acids which occupy pretty much the same site? Okay, and this molecule, arachidonic acid, has to adopt a kind of L-shaped conformation. Now, I don't know, sir is here who is very good in dynamics, he might be able to tell me how a fatty acid molecule which is so long would go like 25-30 angstroms in. What would be the driving force for that? How would it come out like a hairpin? So these are fundamental questions which I had. So you can see that part of it is buried monotopically associated with ER membrane, major place where cyclooxygenase acts as the ER membrane. There are other membranes also it can associate with. So the drug molecules are, in the liver microsomes, for example, in the ER, smooth endoplasmic reticulum. Then the drug has to enter in through the cyclooxygenase site, and the drug, that you have, many inhibitors, we are taking aspirin, we are taking in so many other inhibitors. How do those inhibitors act? And there is a lot of literature which says that ROS are involved. There are several things like hydrogen peroxide and singlet oxygen and superoxide. All these are also there in the cyclooxygenase system. They have been detected by spectroscopy. What is the role of those things there? Are they wasteful, useless products of aerobic respiration, as conventional literature says? So there are minuscule differences. They say that cyclooxygenase 1 has got a smaller pocket, cyclooxygenase 2 has got a slightly larger pocket.
So thank you so much, Professor Venkatesh is here. Hello. Can you put Professor Venkatesh's video on? Is he on? Sir, can you come? Can you give me a video? Yes. Wonderful. Hello, Professor Venkatesh. This is my audience here. Very happy to introduce. Please put his full face here. Yeah. Avroda full face podunga. Yes. Yes, Professor Venkatesh is a chair professor and is a chemical engineer. He's a systems biologist. He's a specialist of the field that we are doing. I had the good fortune of being friends with Professor Venkatesh junior and fellow colleague Sharad Bhartiya. We had, had studied in this IIT Madras. We were in the same hostel, and something that impacted me terribly was my friend's death, and Sharad Bhartiya's friend was also he. So Professor Venkatesh is an expert, and he has MedFlux. He has started this entrepreneurial initiative, and he has 24, 25 people. Am I right, sir? You have 24, 25 people with you. You're working. And he's a very senior professor and very respected, very high-profile professor from IIT Bombay. I've been very fortunate. He was the convener of the first workshop on murburn concept that we conducted at IIT Bombay. Now, I would like to leave the stage to him, and kindly, sir, please give us whatever you want to say. Thank you so much. Please, please, yes, we can hear you very loud and clear.
Okay. Yeah. So I'm supposed to join the first day itself, and I've been traveling to Australia. So I got sort of caught up in the work here. So I'm just thought I will connect sometime so that I can meet up with you guys. So what I will do is I'll just take 15 minutes to tell you, the approach of systems biology and how that sort of connects to what Murli is trying to do in the modern concept, and how the energetics and thermodynamics come together, right? And then one of the approaches that one can take, irrespective of the hypothesis that you would want to put. So I can share it. Let me just share the screen. You're able to see this?
Yes, we are able to see your screen. Yes.
Yeah. So let me start with this video. So essentially, you're able to see the video, right?
Yes, very much. Yes.
Yeah. So this is a trigger, right? The square rectangle, what you have is a trigger, and you can see these cells, following these triggers, right? So it is optically activated, and as soon as that laser falls on it, it will start moving. Okay. So let me just show this again. So this is a immune cell which moves towards a say, for example, if you have a wound, right? So it has to go towards the wound. So what has been done here is that you trigger a signal. So this is optically activated, and then once you have the signal, let me show you another video. Yeah. So you see the trigger comes here, and the cell immediately will start moving towards this signal, right? So that is how sensitive it is. And I change the signal, and it will start moving to the other side.
Wow.
So how is this cell able to respond so quickly to a trigger, right? So you just put a trigger, and then it starts moving. So this looks like a engineering problem, actually, right? So although it is evolved naturally in the system, if you look at it, these triggers have actually come from, although a natural system, you can see that there is some design which is making it respond the way it is. Okay, so in a similar example that we are working on in the lab is also chemotaxis in E. coli. So how bacteria move towards a higher concentration of glucose, right? In this case, immune cells are trying to see where, based on a trigger, chemical trigger, it has to go towards a particular wound location, correct? So for a given trigger, for example, bacteria in terms of a gradient of a glucose, right? In this case, the immune cells, so they are moving towards a particular signal, and then they respond to it. Okay. So if supposing an engineer were to design this, right? So they would, I can design this, with a problem statement that I have to go up the gradient of a chemical, right? So that could be the problem statement. So can I design a system which will take you up the gradient of say glucose or a trigger? So if I have to do that, then I have to have a sensor. I need to measure the concentration of a trigger at a particular location, randomly move to another location, again measure, remember the old concentration, take the difference, and look at that in the positive direction, right? And send that signal to a motor, right? And then it has to start moving in that particular direction. If it is facing in the opposite direction, it has to change the direction and then move in this direction, right? So it's typically what a physicist or engineer would design for a problem statement. Obviously, in this case, evolution has evolved these systems naturally. Okay, these are all Swayambhu, right? So these systems have evolved by themselves to respond in a Darwinian evolutionary pressure. They have evolved this for their own survival. Okay, so now we exactly know how E. coli you need a gradient inside the cell, you need a signal to be generated, you need those laminopodia and all that, to respond to the cell membrane to respond. Okay. Whereas in case of E. coli, you want the flagella to move in a particular direction. So now we know exactly how the sensing takes place, what is the signaling network, how the signal is transferred to a particular location on the membrane, and how the membrane responds either to a flagella or to other mechanisms for the motion to take place. Okay, so this is what I term as the systems biology, where you are looking at a particular characteristic response. Okay, of a biological system, which may be characterized as a phenotype or a very specific characteristic response that you have to a particular input trigger. Okay, so you have input, you have a output, and then now the modern biology has given us all those components, okay, which connect these input to output. Once we understand this, then we have to look what are the design components which are present, how these feedbacks are responding, and then each one we should have a design equation, right? So this is how an engineer would look at and then see how the system operates. So in biological systems, just to set a context, right? So you want to look at right from genotype, transcriptome, proteome, metabolome, and then you have a phenotype, right? And these are hierarchically connections. So what I'm looking at is a external input going to the system. So this is a system definition with the output, but there are inputs which will trigger the system to make a gene, express a mRNA, which will express a protein, which will start a set of reactions or set a signal, and then the cell is going to, the system is going to respond in a particular way. So there is a hierarchical connection of this genotype to phenotype. And I give this example to students where, for example, you have a firefly, so you have a gene which gets decoded, and then you have a reaction which takes place, and then finally the firefly is going to glow. Right? So if I transfer this complete connectivity to some other plant, which was done in 1983, right? This is almost like 40 years back now. You could transfer this complete phenotype to a new plant, right? So the tobacco plant also starts growing because you have introduced the gene, you have made the RNA stable, you have made the protein, you have made the reaction to happen, and then you see that the characteristics has now been transferred. Okay, the response of this plant has also been now transferred because you have transferred this central dogma. Okay? So how do I link all these components and then see what is the design components that are present for a particular trigger and how the system is going to respond? What are the limits, right? Both the flux-wise or thermodynamic limits on the system to operate. So these all become fundamental questions, just like an engineer would design a particular system to understand how it operates, right? Maybe a heat exchanger, or maybe that I'm going to send a satellite to the moon, right? So these are all a particular input that I give and then I design for a particular output to happen, right? So now we understand the physics in such a way that even I can send and make the satellite land on the moon, on the other side of the moon, on the dark side of the moon, which India could do it, right? So in recent times, right? So if I ask the same question in a biological system, obviously it is quite complex. And last 30-40 years, the modern biology has been looking at all these components and how they are connected, which is a massive amount of data that has been generated. So what is given as your genetic, signaling, metabolic network, and they're all connected in a particular way. There is a hierarchical link between cells, different cells coming together and then acting to create a complete system. And bioinformatics and other studies have also added to this knowledge base, right? And complex system to understand and to see how a system is going to provide a particular output for a well-defined input. Right? So what we have been doing in my lab is trying to quantify these systems with the input-output concept, trying to bring all the knowledge that exists, all the constraints that we know about, right from the capacity of the system to how they're connected, what are the feedbacks which are playing a role. So we bring all this data, including omics data, maybe single cell measurements or multiple cell measurements, tissue measurements. So we bring all those and put a constraint and try to see and apply the principles of system science, right, to understand how these systems operate. And that is what is systems biology. I've been looking at multiple systems of design component that exist, that the nature has evolved to give out a particular output. Okay, so just like an engineer would design something, then they will operate, then they would want to control a particular operational space, and if there is any fault, we go and redesign it, and then we evolve, right? So, so an engineer typically takes these steps of evaluating or coming up with a system, right? So they will bring components together and to design and to operate it for a particular characteristic output and how to control it, right? And then you create if there are some faults or it's not working optimally, you go back and then evolve it. Okay. So this becomes a continuous process of evolving a particular system. Biological systems also go through these processes, but in a longer time scale than the evolutionary pressures of survival of the fittest. Some of those components evolve in a similar process. Some of this design, okay. And if for a particular fault or a particular perturbation, if the system is not able to evolve in the time duration that a particular perturbation exists, obviously there are chances that the system may get extinct also, right? Something like dinosaurs that we have seen, that they don't exist now, although we know that they existed long time back, right? So how quickly are you able to evolve to take care of a particular perturbation, right? It comes in the process of that evolution. So I think this example of aeroplane, which is quite a complex system. So we have about more than thousands of computers which are trying to evaluate the pressure on the top, pressure on the bottom, keep it stable, right? Because the physics is, we know the physics of it. The hundreds of feedback loops, there are billions of components. Okay, because this is a bottom-up design, there is a human that designed this from the bottom up. We have a design manual. I know exactly why I have placed a particular component into the system. Okay. Now, pretty much, the takeoff requires manual effort. It's able to handle by itself. It's almost like an autonomous system that, the flight is going to go from one place to the other, and it has to take off, be stable on the air, and then land at a given location. Right? So that is its phenotype, that's its particular characteristic. Okay? And because it's the bottom of design, we have a design manual available. Okay. But if you look at even a simple organism like Escherichia coli, E. coli, right? So the bacteria, which is one of the most well understood systems, right? Although it's a single cell, it's quite complex. It's got about 4,400 genes. Even now, we don't know about all the genes, right? What is their role? There's a lot of connectivity between genes, mRNA, proteins, metabolites. So we said aeroplane has hundreds of feedback loops. A simple bacteria, obviously, you can look at it, there's got thousands of feedback loops. Right? So it's an order of magnitude higher than even a complex aircraft that exists, that humans have designed. So this has thousands of feedback loops. And obviously, this is a top-down system, right? We have not designed the system, right? The system already exists in nature. So there is no design principle available. I don't have a manual for E. coli, right? And we think that, E. coli, there's no competition. You see here, thousands of computers are being used to keep the system stable in the air. E. coli is now surviving in the environment, but it appears that, there's no competition taking place. But obviously, there's a lot of competition that is taking place and making the cell decide, what it needs to do. So, although we feel that there is no competition, in every moment for its survival, a lot of calculation that is happening, and the system is in such a way designed that it is able to survive or it is able to take decisions. Right? So these are all happening through the control, the interactions, that all this connectivity that exists, the complexity that exists between the biomolecules is helping the cell take several decisions. Right? So, like the example that I was giving you, if there is a positive gradient of glucose, it will move towards the higher concentration, right? Because it's the food. So it will search and it will go towards the higher concentration of glucose. So that's how that decision making E. coli has that capacity because it's the design. There is a glucose sensor, there is a signaling pathway, it's got all these components for it to move towards that positive. So a positive gradient of glucose. If I have a positive gradient of an antibiotic or a metal ion, right, which is toxic to it, it will run away. Right? It will be able to sense and then it will say that you move the flagellar anti-clockwise, right? To make the random, movement of the body in such a way that it will start moving away. Right? So it can move towards a positive gradient of a food, but it can also move in the opposite direction of a positive gradient if it is a toxic, like antibiotic or a toxic element. So that decision making is able to do because there are design components inside. Okay? So now we know exactly how these systems operate and how the input signal is translated and how the system changes itself to move towards another direction. Another example is, for example, yeast. Right? You put it under osmotic pressure, right? So you put it under a high osmotic pressure, suddenly, it will sense that, it will start producing glycerol inside the cell, and it will balance the osmotic pressure so that it is able to survive the osmotic pressure till, it makes consistent in terms of its, a certain glycerol composition is built up inside the cell, and so the membrane is stable, okay? Till that time, it will stop cell division, it will just survive. It will be in the lag phase, it will not do anything. Once the osmotic pressure is balanced, then it will start dividing again, right? So you see the inherent intelligence that these systems have evolved through the evolutionary forces that, in order to survive, in order to respond to the perturbations from the environment, these designs have evolved. So can I now, build a manual for E. coli, understanding. See, the player had a very fixed phenotype, right? That is supposed to take off, be stable in the air and land. And it is one of the safest way of travel now because, we understand the physics, even when a turbulence is ahead, the pilot indicates to you, right? The turbulence may come, right? You put your belts, and sometimes they even avoid the turbulence. They'll take a roundabout route so that, they don't hit the turbulence. So all this is possible because we understand the system well. We know the content, we know each component why it is there. Right? And so can I do something similar to these systems? Unlike a fixed phenotype, E. coli has thousands of phenotypes, and depending on the environmental condition, it has to respond in different ways. Temperature stress, osmotic stress, right? There is a toxic element present, other organisms being present, right? A competition is happening with other organisms, so it has to respond and survive. So it has several phenotypes, and for that survivability, we need to have design components present inside the cells, right? The complexity that exists. So that's how, can I now build, if I know all the components, if I know the design principles, if I know how for each of those phenotypes the cell is responding, okay? And there's a lot of quantification, numbers involved, just like in engineering system, right? It is just not that I have to bring two components together. I need to bring two components and then understand what is the operating values, right? So I can have a circuit, but I need to know what is the resistance, what is the capacitance, what is the operating constraints on these components, right? That I should understand. What is the binding constant of a protein, right, on the sensor? Like, for example, glucose binds to the membrane. So what is its binding constant? All that plays a role in actual functioning of that particular system. So you need to understand, this is almost like writing a manual now for E. coli, okay? For each of those phenotypes, given all the components, given the connectivity, right, and given the well-defined input, how the system is going to respond. So can I build a manual? That's what a systems biology, trying to see how all this operates. Obviously, these components are complex because the input are many, there's a lot of feedback inside, each component can interact in a different way, and then you have a different outputs that has to be analyzed. So, what is this complex system? Right? So, how do we even define this complex system? So, let me just give you one example. Right? I'll just take another 10-15 minutes to tell you, and then I'll stop. So, for example, I have a blue ball and an orange ball. Okay. Now, supposing if I randomly pick one of the balls, what is the probability that it's a blue ball? Obviously, it's 50%. Okay, but I'll slightly change the experiment. Say, for example, if I pick a blue ball, in the next step, I'm going to add another blue ball. Okay, and then go on doing it. Right? So that is the experiment. The experiment is I'll randomly pick one, but I will not just stop there and then independently, mutually exclusively do this experiment. What I'm going to do is, whatever color ball I pick, okay, I'm going to add the same color ball in the next step. So if I do this 50,000 times, right? So I'll have 50,000 plus 2, right? 50,002 balls I'll have at the end of the experiment. Now I ask you at the end of this, what is the probability that it is a blue or a red ball? Okay, I hope you understand the experiment that I'm doing.
Yes.
Okay,
Yes.
so if I do this, then obviously I cannot predict the probability, right? I always start with a 50% blue and red ball, but then I can have any probability, right? Going to a very small probability to a very high probability and
Yes.
to the 50,
Yes.
right? So it all depends on the bias that I'm giving in the initial step.
First.
Supposing I have only the blue balls, right? Then the probability that I have a blue ball will be very high. If I go on putting red ball, then the probability of red ball will be very high. I can go alternative, blue, red, so then I'll be 50%. Okay? So I can go to any of these steps. So this is what is the complexity. I've created one non-linear step in the experiment. So what it has done is it has given multiple outcomes and it is dependent on history. Right? So the outcome is dependent on history. So this is what is the basic concept of complexity, that it depends on the history, okay, and it can go to many states, multiple states. So we all start with a single cell at conception, okay. So they are not very, very different. Right? At that stage, we are mutations and steps that we carry from our forefathers is what is the difference that we carry, okay, which may be less than 0.1%. So we all start with almost similar system, but we all end up different, right? So now we are all very unique outcomes that we have come out, although the system was very, very much similar. Okay? That is because our own history is different, our steps that we carry is different, our own experiences when we are growing is different, right? So all this puts you in a different steady state and we all become unique. So this is a classic example of complexity, right? which can put you in multiple states depending on the history that you take, okay, or the experience that you have had, that will take you to a new steady state. So this is the challenge, right? So although we say equalize one system, but each equalizer has its own journey that they take, and it has a capacity to go to several outputs. So that is one of the reasons why bacteria also picks up antibiotic resistance, because there are some which escape the effect of antibiotics just because of the complexity that exists, right? So people did some experiments and they found out that something like 5 to 10 cells in 1,000 cells escape the antibiotic effect, and they get the chance now to mutate and then get resistance, over time. So these kinds of complexities and the chance for evolution, right, happens only because of the complex system that exists, different components which have come together and then evolve with the evolutionary forces, these designs which have come. Okay, so just like engineering systems, we need to look at multiple steady states, initial conditions, parametric sensitivity, the bifurcation, robustness, adaptability, evolvability. At some level, you need to adapt to inputs. At some other level, you need to come up with completely new design and evolve. Right? You need to be robust in some instances. Right? In some instances, you need to adapt. So these components are inherent in the system, and depending on how the perturbation occurs, the system evolves to give robustness, adaptability, evolvability. And then to evaluate this is what a systems biology. How do you put it in this perspective, right? So we look at nonlinear dynamics, multiple feedback loops, multiple interactions, cascade structures, feedforward loops, interaction between modules, time scale separations. We need to look at all these aspects when we are writing a manual for a particular phenotype, and then put it in that perspective, right? So only then we understand truly how a input gives rise to a very well-defined output, okay, in a context of the system, right? How the systems are going to work. Obviously, all these are limited by thermodynamics, limited by rate, the capacity of the system, and it can perturb at many levels in terms of the feedback, and that is why these are complex systems giving rise to very non-linear outputs, right? Which is very difficult to figure out just by the component interaction. Right? It is very difficult. Just if I give a map of the network, it's very difficult to say what exactly would be the output for a given input, because you need to look at all these dynamics. Right? So there are, for example, I don't know, in Kerala there is a flowering, a plant flowers once in 12 years. Typically, I think in the February, March, in a period of 2 to 3 weeks, they flower. Okay, once in 12 years. So how is the system able to do that? Right? So it has to keep track of the time, not just in minutes or whatever, but decades. Right? The hormonal response should happen, and precisely, 12-year time scale, okay, but it has to do, photosynthesis, it has to do respiration, all that, in a daily basis, right? But the flowering should take place once in 12 years. So that's how the system has evolved. Right? So there's a time scale separation. There's different components operating at different time scales. So all this is possible in such complex systems. So we do look at different network motifs, what kind of outputs they give, and then sort of bring it together to show how a particular system responds. Okay. So I don't have time to go through many examples, but typically what we do is we understand all the knowledge base that exists for a particular system. We bring it down. We put all the constraints, right? Stoichiometry, thermodynamic capacity, because we still are not sure whether we understand completely, right? So we know half knowledge is there, but we cannot sit quiet for the complete knowledge to be available. So we take this unbounded solution space. We put in all the interactions, the topology, the different connectivities. We bring it together. We look at what is the data that is available in terms of stoichiometry, versus thermodynamic limits, in terms of the capacity, the rates. So we bring all that together, and then we apply like what Arthur Conan Doyle says in Sherlock Holmes, right? That, how often have I said to you that when you have eliminated the impossible, whatever remains, however improbable, must be the truth, right? So systems biology heavily picks on this philosophy that you understand the knowledge, you bring in all the components, you bring in all this network, and then you put all these constraints, right? So whatever is known, that it could be omics data constraint, it could be stoichiometric constraints, it could be thermodynamic constraints, right? So we bring in all these time constants, all these constraints that are known, I bring it down, and then in this unbounded space, I look at where I'm landing, right? So because of all these constraints and topology, where do I land, right? And that becomes the probable truth, right? So that is how we try to bring in this input-output concept and then say how a system is going to operate and how I can build a manual for biological systems. Right? So this is how there's a lot of mathematical modeling, bringing data together, mechanistic details of feedback, which interactions I need to neglect for a particular phenotype and which I need to account for given the time scale.